Hi everyone, welcome to the second UlyssesNeuro Podcast on Fragile X. In the previous one, we talked with the Irish Fragile X community to focus more on patient and family perspectives. And in this one, we're going to go more in depth on the research side of Fragile X. I'm Carol, and here with me are John, Charlotte, and Max from our lab, and Matthew Campbell. Hi Matthew.
Hi, Carol. How's it going?
Good, thank you. And can you tell us who you are and why you're joining us?
Yeah, so I'm a principal investigator based in the Smurfit Institute of Genetics in Trinity College. And I've been running a research lab since about 2013. And the major focus of my lab is on understanding the role of the so-called blood-brain barrier in health and disease states. And in 2016, I started to work with a group based in the College of Surgeons, RCSI, here in Dublin. And they were beginning to form a concept of a Science Foundation Ireland funded centre that was called FutureNeuro. And they invited me to become a principal investigator within their centre. And so really that a lot of our interest in Fragile X syndrome and our interest in working with Ulysses Neuroscience is founded on the basis of this SFI funded centre, FutureNeuro, and trying to use the skill set that my lab has developed over the past few years to see how we could apply it to Ulysses Neuroscience's outlook on treating and exploring Fragile X syndrome.
What is FutureNeuro exactly for any listener that might not know it?
Yeah, so about 12 to 15 years ago, Science Foundation Ireland began to invest heavily in what they termed centres of excellence. And so largely they've invested a lot of money in these centres whereby industry come along and can put up 50% of the funding. Science Foundation Ireland will then release another 50% of funding. And the concept was that they would build centres of excellence in ICT and engineering. And really there wasn't one, there was a gap there that they hadn't really funded a centre for the life sciences. And so in 2016, they invested heavily in this new SFI centre called FutureNeuro. And its aim essentially was to try to join academia and industry together to explore novel therapeutic strategies for treating rare neurological conditions like epilepsy and ALS, so that the primary focus at the moment for FutureNeuro is epilepsy and ALS. But the goal moving forward is that they expand into other neurological conditions. And again, it's working on this concept that they want to bring academia and industry together to co-fund projects. They're interested in biomarkers, they're interested in developing therapeutics, and they're also very, very interested in the likes of electronic patient records, this EPR, and being able to accurately identify rare genes that cause these rare epilepsies that patients mightn't ever get the chance to have a genetic diagnosis of their disease, that they're really trying to hone in on that and focus on developing this electronic patient record that could allow for this genetic diagnosis to be possible in the years ahead.
And John or Max, why is Ulysses Neuroscience interested in Fragile X, and why are you joining with FutureNeuro? What is the research that you carry on this disorder?
Carolina, I can actually tell you how that started with Fragile X syndrome. We were initially not interested in Fragile X syndrome at the beginning of Ulysses Neuroscience, and the interest was developed because we were already working on another neurodevelopmental disorder, which is a rare brain disorder linked to alterations and mutations in a gene that is in the X chromosome, so very similar to what is happening with Fragile X syndrome. And when we then got into connection with the Fragile X Irish Society here in Ireland, and we started to look into Fragile X, we saw there was some overlap in the biology of the two disorders, so the CDKL5 deficiency disorder on which we were working, and the Fragile X syndrome. So we saw some connections in there, and this is when we started to get interested in Fragile X syndrome and in trying to understand the biology and the pathogenesis of the disorder. And so we started talking with pharmaceutical companies, and we also observed that there was a lot of interest, I would say, on their side to try to understand more about the biology and also to develop new treatments to cure the disorder, so disease-modifying treatments. We decided to really go into the field, and we established our own colony of an animal model of Fragile X syndrome, and John will tell more about that, which are knockouts, essentially are animals that lack of the gene which mutates in Fragile X and gives actually the symptomatology that belong to the Fragile X syndrome. And since we want, and this is one of our core values for Ulysses Neuroscience, to work with the academia, to do things together with academia, we also saw a potential connection between what we were doing in terms of our own research, which is around synaptic plasticity, and something that we will talk later, which is called microtubule dynamics. And we saw there was some connections with the blood-brain barrier. And this is when we come to talk with Matt, try to make use of their expertise to integrate it in our project. And for us, it's very important to work with the academia. And I guess John, as academic liaison manager, can say a little bit more about that.
Thanks, Max. Academic collaboration is indeed key to Ulysses Neuroscience. We like to work with academic groups as well as obviously industry, because it allows us to really harness the full potential of neuroscience research by drawing on the best of every situation. I also like academic collaborations because they tend to be quite organic in how they come about. This is especially true in this case, as I used to be a postdoc in Matt's lab. And there I was involved in a project looking at the blood-brain barrier in schizophrenia, and we got some really nice data on that and published really well. But I was always interested in exploring the role of the blood-brain barrier in other psychiatric disorders. And it was just serendipity when I arrived at Ulysses Neuroscience, and Fragile X was one of the key research areas. And this is one disorder that I had actually thought this could be rich pickings for looking at the blood-brain barrier. There's very little known about the neurovascular system in Fragile X, and indeed very little known on how this might relate to the known problems with synaptic plasticity, the behavioral changes, the cognitive changes that we associate with the disorder. So this project with FutureNeuro and with Matthew Campbell is part of a strategy at Ulysses Neuroscience, not just in Fragile X, but across a number of disorders, to improve the translational validity of the mouse models and the preclinical models that we use. And this includes everything from molecular targets, biomarkers, through to behavioral paradigms, and it's all done by refining our research in light of the discussions we have with patient groups like the Fragile X Association in Ireland.
Before we get into the real nitty-gritty biology of Fragile X syndrome, I think it's really important to note that the current treatments for Fragile X are limited to addressing the individual symptoms only. These symptoms include mood problems, seizures, aggressive behavior, and ADHD. So through our research in Ulysses with people like Matt or with pharma companies, we hope to target the core biology of Fragile X, to understand at a cellular level, both in neurons or in other cells in the brain, how cellular signaling is disrupted, and in a way that leads to the development of Fragile X and the associated symptoms. So by identifying the underlying biology, we hope to find new targets to develop treatments for Fragile X as a whole, and not just for individual symptoms. And this is where animal models really come into research. We can use models to study brain signaling mechanisms in a way that we can't with people or with patients. So I think this leads to John and Matt maybe talking a little bit more in detail about our joint project and the Fragile X animal model that we're using for this study.
So you mentioned the genetic origins of Fragile X quite a few times. Can you give us an overview of what Fragile X is and some background on the genetics of it?
No problem, Carol. I'm quite happy to talk about this. Fragile X syndrome is a genetic disorder, and occurs in individuals where there's a mutation in a gene called FMR1. It's one of the most common single gene disorders in humans, and is one of the leading causes, in fact, of cognitive disability in terms of inherited cognitive disability in humans as well. So it's a fascinating disorder to look at, not just in terms of what can we do for patients, but also in understanding the brain itself. So FMR1, this gene involved in Fragile X syndrome, it codes for a protein called FMRP. FMRP, it's involved in the creation of synapses, which are the connections between neurons during brain development. So when your brain is developing post-conception, your body starts developing, you get these neurons forming, you get these other cells forming in the brain, glial cells, you get the vascular components of the brain forming, and the neurons have to wire up to each other. And FMR1 seems to be quite important in this process. In the older brain, it's also, and by that I mean kids up, not just older individuals. It's involved in a process called synaptic plasticity. So this is where you have all these synapses connecting your different neurons, and they can change in how strongly connected they are to each other. And this is what we believe is the biological basis of memory formation. This makes sense with what we know of fMRP from a molecular level. At a molecular level, fMRP regulates synaptic plasticity because it acts as a taxi for the essential components for synaptic plasticity, in that it takes the mRNA that's produced by the nucleus of the cell, of the neuron, and it taxes it to the end of the dendrites where it's needed to form or change the synapse. And here at the dendrite, the molecular machinery that's there will turn these mRNAs, which are transported by the fMRP, into a protein. In Fragile X, this process is impaired because individuals cannot produce the Fragile X protein at all, fMRP. The reasons for this is because the FMR1 gene, which codes for fMRP, cannot be translated into a protein. And the reason it can't be translated into a protein is because it has these, what we call genetic repeats. Basically, in the genetic code that's used by the body, you have the CGG that repeats over 200 times at the beginning of the mutated form of the gene. This prevents the gene from entering into the cellular machinery that I mentioned a moment ago for protein production. So in Fragile X syndrome, you cannot produce the protein because the gene has basically got this big, huge loop at the start of it, which kind of clogs up the machinery when it tries to enter. You can think of it as like when, if you have an old-fashioned cassette or VHS tape, that when it goes into the machine, if it gathers up in the spools, it will jam the machine and won't go any further. Or a paper jam in a printer, or too much material gathering up in a sewing machine. That's probably enough analogies there. The knock-on effect of having no fMRP is that then your components for synaptic plasticity, which are produced in the center of the cell essentially and have to get to the tips of the neuron, they no longer have a taxi, so they can't get from where they're transcribed to where they're translated into a protein. And this means then that your ability for synaptic plasticity is then reduced. This fits with what we see in the mouse model. So in mice, we don't introduce this repeat at the beginning of the gene, which causes it to jam in the machinery. Instead, we just take the gene out entirely. So this is called a knockout. And in the FMR1 knockout mice, they indeed don't produce fMRP because they don't have the gene to produce it. And they have impairments in synaptic plasticity when we look at it on an electrophysiological level. And they also have impairments in memory. So they find it hard to learn new things. And it really pairs really well with what we know about what fMRP does in synaptic plasticity.
This leads us on to our project that we've started with Matt and FutureNeuro. We've named it Proteus after the Greek shapeshifting sea god. So for those of you unfamiliar with Proteus, he appears in two main kind of literary inspirations here at Ulysses Neuroscience. That's Homer's Odyssey and Joyce's Ulysses. In the Odyssey, Proteus is described by Menelaus, who's a veteran of the Trojan War, in a story he tells to Telemachus, who's Ulysses' son. When Menelaus was on his way back from Troy, he was blown off course by storms. And the reason this was, of course, because he didn't please the gods with enough ritual sacrifices. He didn't know which gods he offended, but he was told that if he was to capture Proteus, who is a son of the gods, he'd be able to tell him which gods he offended and how to make things up to them so he could get back home. So to catch Proteus, who's a shapeshifter, they snuck up to him on the beach while he was counting seals. They actually were disguised as seals themselves, and he counted them amongst his pet seals. And they jumped out and caught him, and then started to wrestle him into submission. And at this point, Proteus did what he does best, which is change shape. So he turned into a lion, a dragon, a leopard, a boar, and then into like water itself, and finally into a tree. They held on all the way through and finally defeated. Proteus then did give up the information that Menelaus needed to continue his journey home. So this ability to rapidly change shape, and we can see as plasticity, much like we talk about plasticity in the brain, it's our metaphor for these changes that we see in the brain. So the changes we see in microtubules at the molecular level, the blood-brain barrier at this kind of tissue level, and synaptic changes between neurons at this functional level. By holding on to this plasticity and forcing it to give up its secrets, we hope to make a meaningful difference in the lives of individuals affected by Fragile X syndrome.
That was a really nice introduction to Proteus, John, and to where our origin, the name Ulysses, came from too. So our project within Proteus is really based around Fragile X and the blood-brain barrier. So maybe, Matt, you could explain to our listeners a little bit about how we think the FMR1 protein and gene may contribute to an alteration in the blood-brain barrier integrity, and how this may impact on the syndrome severity and even the development of new treatments.
So we're coming at this from a slightly different angle in that my lab focuses exclusively on the blood-brain barrier. So we generally don't focus on one disease in particular, and we allow our research to dictate what diseases we work on. And in the context of Fragile X syndrome, the fact that individuals are developing neuropsychiatric phenotypes and seizures in some cases, depression, anxiety. About 4 years ago, we serendipitously discovered that the blood-brain barrier seemed to associate very, very strongly with animal models that went on to develop these neuropsychiatric-like conditions. So when we develop models in the lab, transgenic and knockout models that allow us to control specific elements of the so-called blood-brain barrier, so the lining of the blood vessels within the brain, if we make those blood vessels leaky, we can gradually induce a whole plethora of phenotypes in animals that recapitulate a lot of human diseases. So that's kind of where this started off. And that's why I've got an interest in psychiatric disorders, got an interest in epilepsy. That's what's aligned a lot of my research with FutureNeuro. Because it now appears that the blood-brain barrier and a dysfunctional blood-brain barrier appears to be associated with a whole range of different neurological conditions, not just Fragile X syndrome, not just epilepsy, but schizophrenia, bipolar disorder, anxiety. And we're developing a lot of data to suggest that is the case in both animal models, but also in clinical research that we're undertaking. In the context of how does the FMR1 gene and the triplet repeats that John mentioned that are disease causing, how does that cause, or does it cause blood-brain barrier disruption, or will it cause blood-brain barrier disruption? The answer is we simply don't know, and that really is a large part of the driving force of Proteus, that we initially want to characterize these knockout mouse models and to see whether or not the blood vessels in the brain are intact or not. If they're not intact, to what level are they disrupted? How leaky are they? And then what we want to do is we want to try to titrate that leakiness. So if we can show that these blood vessels are leaky, well, how does that align with the phenotype and the behavior that these mice are showing compared to littermate controls, wild type normal controls? If we can do this, what we're essentially doing is we're opening up a whole range of other therapeutic targets. So yes, we know that the FMR1 gene is causative. It is the causative factor, as Charlotte has mentioned. It drives the biology and the pathology. But from that initial seed of pathology, there are likely going to be numerous points during the development of the disease that we can potentially therapeutically target. And essentially that's what Proteus is about. From my lab's perspective, we want to know, are there targets within the blood-brain barrier that we can prevent the barrier from being leaky, and will that ultimately prevent the negative sequelae of the disease? So that's kind of where we're coming at it from. So maybe not so much that the FMR1 gene and the protein product FMRP is actually going to cause a disruption of the blood-brain barrier per se, but that downstream of that pathology and that pathological protein and the synaptic dysfunction, as John has discussed, we are hypothesizing that will eventually lead to blood-brain barrier disruption and possibly blood-brain barrier breakdown. But excitingly, we do know that the blood-brain barrier is therapeutically targetable. So we can restore the barrier's integrity, and the concept will be that could we actually treat some of the negative sequelae of Fragile X syndrome by targeting the blood-brain barrier.
I think it's important for everyone to know the FMR1 gene is expressed in every cell in the body, and so it has a role to play in those endothelial cells which make up our blood-brain barrier. Another important aspect of the Fragile X condition is the movement of the FMRP protein, which is encoded by the FMR1 gene within the cell. And it's this movement from the nucleus to the points of the cell where the FMR1 protein is actually functional is really important. And it does this through interactions with microtubules. Microtubule dynamics is an area which Ulysses is very interested and has great expertise. And I think it might be nice, Max, if you could give us a little introduction to microtubules and how they relate to CNS disorders and then to Fragile X and the FMRP protein.
Charlotte, thank you for the question. And as you have mentioned, microtubules are indeed in between the main expertise of Ulysses Neuroscience. I personally have been working on microtubules for the last 20 years, and even my PhD was focused on investigating the role played by microtubules in the pathogenesis and treatment of major depressive disorders. So, what are the microtubules? Well, the microtubules are cellular organelles. They are in each single cell in our body, and they play vital roles in there. They're really fundamental. They're part of the so-called cytoskeletal triad, which is composed by microtubules indeed, and then the actin filaments and the microfilaments. As I've said before, they're vital for cells. And, one example is the mitosis. And this is also when and students in the biology field, they encounter microtubules, and they normally remember microtubules for this reason, for the mitosis. So microtubules form the mitotic spindle, the so-called mitotic spindle in cells that are separating, that are producing new cells. And the mitotic spindle is fundamental for the correct displacing of the chromosomes. So it's without mitotic spindle, there cannot be production of new cells, and so essentially the body would not exist without microtubules. So this is just an example of a very important and vital role played by microtubules. If you go a little bit more into the biology of microtubules, well, they are composed by an heterodimer, which is a protein composed by two different proteins. This is why it's called heterodimer. And so those two proteins are the alpha tubulin and the beta tubulin. They are linked and they form this heterodimer that then polymerize into so-called protofilaments, and then the protofilaments assemble to form real tubular structures that we can clearly see with the electron microscopy and inside each single cells. And they're very fascinating images, by the way. And they're real tubes. I mean, they have a hole in the center. We don't know what happened actually inside the hole. And this is one of the huge mystery of the biology of microtubules. So there are some investigations in process, but, the technology available is still not enough to understand what happens inside the hole. But on the surface of microtubules, there are a lot of things happening. We know them pretty well, I have to say. And in neurons, what they do in neurons, because neurons are important for us. Those are the cells that we are going to study in Proteus, and Fragile X syndrome is a disorder of the brain. In neurons, they are important because of their characteristic of being dynamic. So they are dynamic polymers, so meaning that they continuously alternate phases of elongation with phases of shortening. And this is the way in which they can control the different functions inside the neurons. However, this microtubule dynamic is different in different compartments of the neuron. As we know, neurons are formed by a body where we have the nucleus that contains the DNA, and in the body there are also other organelles which are have been designed by nature to create the elements that the cell needs, like proteins or other elements are actually produced in the body of the cells, and then they are transported in the other parts of the neurons, and they are transported also to the end of the neuron in the axon, and then the axon is the other part of the neuron, where the transport of those elements actually happens, and some of those elements have to reach the end of the neuron, so they have to reach the end of the axon where they can be released and they can interact with the receiving neuron or with other cells, which we do have in the brain, that can be the glial cells, for example, that support the neurons, or also endothelial cells, as it is the case of the blood-brain barrier. So in the axons, those elements are transported via a track, and the track is indeed formed by microtubules. And the track has to be very stable. So despite microtubules are very dynamics and they need to exchange with new tubulin to keep, the kind of freshness of the track. Anyway, the dynamic has to be very, very low in the axons, and this is because the track have to be stable, to promote the transport of those elements up to the end of the neuron. The other part of the neuron are the dendrites. And the dendrites are actually those compartments of the neuron receiving the elements that are transported via the axon. Indeed, when the axon releases those elements into the so-called synaptic space, they're then received by the dendrites of the receiving neurons. But dendrites have another important role to play in the brain because they're very plastic. What does it mean? It means that they change their structure very, very quickly, and they can produce some new branches which are used to make new connections with other parts of the axons or with new axons from other neurons that are close. They also remodel their structure very often, and this is also to maintain correctly the connections with the neurons. They do this very, very quickly, and they can do this only because the microtubule dynamics is really high in there. And this is what promotes those processes of synaptic plasticity. So in an healthy neuron, it's a question of balance between low dynamic in the axon to promote the transport and a high level of microtubule dynamics in the dendrites to promote synaptic plasticity. Now, what all this has to do with Fragile X? Well, we go back to the now famous in this podcast FMR1 protein, which is the one mutated in the Fragile X. So FMR1 needs to be transported via the microtubule tract to interact very likely with other cells, including the cells that form the blood-brain barrier, or to somehow promote the integrity of those cells, so to promote the vascular integrity. So a microtubule dynamic which is correct in the axon is important for allowing the FMR1 protein to play its role. On the other hand, so in this case in the dendrites, we know, and this has been shown by other researchers, by other colleagues, that the FMR1 protein actually interacts with one of the so-called microtubule associated proteins, which are proteins that interact with the microtubule surface and modulate their dynamics. And the FMR1 protein interacts with one of those proteins, which is called the MAP1S, which modulates microtubule dynamics in the dendrites. Essentially, MAP1S makes the microtubules more dynamic in dendrites and promotes their plasticity. So the role it's important. And we do believe that mutation FMR1 protein that we have in Fragile X patients may disrupt the interaction with MAP1S. And this is the reason why we do believe to have altered synaptic plasticity in Fragile X patients. And altered synaptic plasticity means a lot of alterations in social behavior and in processing of stimuli which are related to emotional behavior. So we really believe that these alterations are important for Fragile X syndrome. And we also collect some pilot data in our lab by using an animal model of Fragile X syndrome, which is the essentially mice lacking the gene of expressing FMR1, so they don't have the FMR1 protein. What we have seen in those mice is that we have huge, massive alterations in the brain concerning microtubule dynamics. So those data were very promising for us, and this is also one of the reasons why we decided to engage into Proteus as a joint project with Matthew.
Thanks, Max, for that introduction to microtubules and how they are essentially altered in Fragile X and contribute to the disease. I guess a really important question to address within this podcast is how we can translate all the information we get from our animal models and from the lab, and how we translate that into a clinical setting so it can have impact for our patients. Firstly, I'll ask you, Matt, in relation to our project on the blood-brain barrier, and then to you, Max, how our findings on microtubule dynamics in the lab can translate into the clinic.
Yeah, so from the perspective of blood-brain barrier biology and how we examine how intact the barrier is in human subjects, we have numerous tools available to us and that my lab uses routinely in collaboration with clinical colleagues. So one of the main ways that people would look at the integrity of the blood-brain barrier in human subjects is to screen the systemic circulation, so serum or plasma samples, for molecules, proteins that are really only found in the brain. So molecules like S100 beta, neuron-specific enolase, phosphorylated tau, GFAP. These are molecules that are present at very, very low levels, if even detectable at all in the blood. And if they are detectable in the blood, it means that there is, at some stage, there has been a compromise of the blood-brain barrier, or there is an ongoing compromise of the blood-brain barrier. So that they are useful tools. They have certain utility. So their utility is that they can tell you whether the barrier is open or not. They're not brilliant at giving prognosis to patients. So they're not great at aligning whether or not the disease course is going to be worse or better. What we also need to make use of is imaging, essentially. So magnetic resonance imaging. One of the key tools that we have for clinical research is a technique called dynamic contrast enhanced MRI, DCE MRI. And what this allows you to do is, while the individual is lying in an MRI scanner, we can inject a contrast agent or a dye that's gadolinium based. And essentially this dye is highly negatively charged, so it won't passively diffuse from blood into the brain. And so in a normal, healthy brain, you'll see an increase in signal as the gadolinium dye floods into the vasculature of the brain, and gradually the signal will dissipate as the gadolinium gets washed out of the brain. If there is any compromise at all in the integrity of blood vessels within the brain, the gadolinium will seep from the blood and deposit into the brain, and you'll get this residual signal that we left. It won't wash out, and that will give you a very clear and accurate indication as to whether or not the blood-brain barrier has been compromised. But obviously, using MRI, it also allows you to see the site specificity, because the blood-brain barrier isn't this homogeneous entity. It differs in different parts of the brain. Some parts of the brain are inherently very, very leaky, like the hypothalamus, and other areas, cortical regions, are incredibly tight, where you'd never expect to see a gadolinium signal. So they're the two main tools that we have available to us that could rapidly translate animal findings into real and meaningful data from clinical research.
So thank you, Matt. And I will take it over from here just to explain also the translational relevance of the microtubule endpoints that we are going to analyze in Proteus. And I also like to underline the fact that this is really important. It's for each research project involving animals, so preclinical projects, it is very important that the endpoints have a translational relevance, meaning that they can be then analyzed correctly into clinical settings, so in the patients, in the human beings. And this is one of the core values for Ulysses Neuroscience as well. I mean, translational research is what we do, and it's the only way to advance correctly what we discover in the animals, then into the humans, and this will promote, of course, the advancement of drug discovery and discovery of new treatments. So what can we do with microtubules? Well, the same proteins that we use to analyze the dynamic of microtubules in the brain, we discovered to be detectable in plasma of human beings. So this is very, very important, and we have now collected a series of data and number of results showing that in brain disorders associated with altered synaptic plasticity, those microtubular proteins are altered in the plasma, and they're altered in the same way in which we can observe them altered in the animal model of that specific disorder. So this will be our way to translate the endpoints into the humans by looking into the plasma of patients and by analyzing the same microtubule proteins that we have analyzed in the brain of the animals.
Before we wrap up, I just wanted to highlight the importance of collaborations for us, Ulysses Neuroscience, with academic experts like Matt and also with pharma companies which have a great interest in Fragile X. These collaborations are essential to advancing our scientific methods, so we have a deep understanding of disease mechanisms, in this case Fragile X. And it's not through one of these elements, but a combination of all our efforts, we can make advances in the field and have a real impact which benefits patients. Because at the end of the day, this is why we all got into research.
Sorry, I totally agree, Charlotte, that I alluded to this at the beginning, where I said we need to go back to patients and see what is going on with them, and especially with these new biomarkers and potential mechanisms in mind. Nobody has really looked at it in Fragile X patients. Nobody's really looked at microtubules or the blood-brain barrier, so it's important to go back to them and see how do what we know about the biology of these things fit with their symptoms and with their needs, and how can we bring this back in order to do better research preclinically in order to expedite what we find or what we know about these processes into therapies or into potential assays that we can use quickly, not wait another 20 years. Because I mean, the problem with industry can be that sometimes it can be a bit cutthroat. They don't see an immediate gain. It's forgotten. The problem with academia is you could have an interest in something and spend 20 or 30 years working on it, but not get very far because you're one group with one question in mind. So I think it's good to kind of bring these together and find the overlaps that we can really push these interesting questions and really push pharma to consider these as important approaches. And ultimately for clinicians and patients to get the best medicine possible for these disorders.
So thank you everyone for participating in this podcast and giving us great insight on the more scientific side of what Fragile X is and what our project Proteus will be. And thank you everyone for listening, and please subscribe to our channels where you can find our previous podcasts and keep your eyes peeled for the next podcast. Thank you. Bye.